Hello, everyone. For this session, we're very happy to have Dr. Julien Camperi to share with us about his research on CRISPR Cas9 RNP complexes. Dr. Julien Camperi received his Ph.D. in Analytical Chemistry from the Paris Sciences at Lettres University in France. He's currently a scientist at Genentech in South San Francisco, California. He supports and provides technical leadership throughout the analytical development process in the T-cell therapy engineering group, moving novel therapeutic molecules from research to clinical trials through collaboration with cross-disciplinary teams. Without further ado, let us welcome Dr. Julien Camperi.
Thank you, Daniel, for this introduction. So today I will talk about CRISPR Cas9 and more specifically about physicochemical and functional characterization of different engineering Cas9 variants, and also go to further and discuss about CRISPR Cas9 RNP complexes. So for the outline of this presentation, I will start with a background on CRISPR Cas9 gene editing, so how it works. Then the first part will be about all the different UHPLC and C-based methods that we can use to do a full characterization of Cas9 protein. So we will see how we can determine the purity, the biological activity, how we can determine the isoelectric print, and how we can use mass spectrometry to have some information about intact mass, or even how we can do some peptide mapping on the Cas9 proteins. And for the second part of this talk will be about physicochemical and functional characterization of RNP complexes. So what is a CRISPR Cas9? So is a powerful tool to precisely manipulate cellular behavior and function. So how it works? So CRISPR Cas9 system consists of a single GAIN RNA that you can see here in red, and Cas9 in blue, and the two different species can form something that we can Ribonucleoprotein RNP complex. And then this complex can bind directly to the target DNA sequence complementary to the GAIN RNA. And following this action, we can generate a double strand breaks. And following that, we have DNA repair mechanisms. And for example, we can introduce exogenous repair template, and we can have a modified locus. So this could be potentially valuable for T-cell therapy or all these kind of applications. So the first part of my talk will be about all the different methods that we can use to characterize the Cas9 variants. So the Cas9 endonucleotides from Streptococcus pyogenes is the most widely used in the CRISPR Cas9 system. But over the last year, we have some effort to minimize off-target cleavage. So basically the unwanted mutation that can cause by CRISPR Cas9. And for that, we have a lot of ongoing effort to develop some engineering Cas9. Recently, Vakulskas demonstrated that only one single point mutation in the wild-type SPCas9 can maintain a high target activity, but also a very low level of off-target mutation. And this is particularly very interesting for clinical application. And in addition of that, the CRISPR Cas9 system needs to have a very high quality gene editing reagent, which is very critical for patient safety and is the expectation by regulatory agencies. So, as I mentioned, CRISPR Cas9, we have two different entities. We have single GAIN RNA and Cas9. And as you probably know, in the different field, antibody, small molecule, UHPLC, and C are the technique of choice to assess the chemical purity of whole drug substances and drug products. So the first part here will be about the GAIN RNA. So at Genentech, we have two main methods. The first analytical strategy is for purity assessment of the single guide RNA. So in order to do that, we employ ion pair reverse phase chromatography at high temperature in the presence of organic co-solvent. And you can see here on this figure that if we do some stress condition, you can see control versus over time, we can generate some impurities. So basically with this method, we can conclude that it's also stability indicating because you can see the presence of shortmer, meaning impurities over time. And we can also, the same thing with acidic conditions. So then we have also another approach, or we need to do the sequencing of the single guide RNA. So usually people can use single sequencing or NGS, but here we also use HPLC via parallel ribonucleotide digestions. And so you can see here one example, or we have different chromatograms of different guide RNA, and basically you can determine all the different impurities, and we can have a high sequence coverage and be sure that we don't have any problem with the single guide RNA, and it will be very specific to our target. So then I want to discuss more about Cas9 because so far we don't have any method of sufficient quality for use in regulatory application, and especially for the newly modified variants. So at Genentech, our strategy is to, first of all, to determine the purity data. We suggest to use CSDS method. So basically, this method is largely used for antibody characterization, for example. So here you can see that we took 3 different Cas9 variants with SA-Cas9, SP-Cas9, and SPY5-Cas9. So SPY5-Cas9 is the high fidelity variant, meaning that it's this one with one single point of mutation. And you can see here that if we do the expanding view, we have some peak before the main peak, corresponding to the low molecular form, and after the main peak, we have some high molecular weight form. And so in this case, we saw that we have a very good purity with all the different Cas9 variants are the purity superior to 91%, and the SPY5-Cas9 is the highest purity with 97.1%. Now, the second question is to know if our method is a stability indicating method for Cas9. And in order to do that, we find some stress condition with some temperature. So basically, we eat the guide RNA at 30 degrees for 2 weeks or 4 weeks. And you can see if we compare with the control that we have significant increase in impurities of the low molecular forms and also for the high molecular weight forms. So basically, this method is also a stability indicating method and can be used to indicate the stability of Cas9. So now the next essay is to how we can determine the biological activity of Cas9. So in order to do that, we need to put Cas9 in the presence of single guide RNA and of DNA substrate. So here you can see one example. So on the first figure, you can see that when we analyze the uncut double-strand DNA using bioanalyzer 2100 electrophoresis system from Agilent. And you can see that when we just inject the uncut double-strand DNA, we have only one peak and 2 different markers. So then if we decide to put a specific Cas9 RNP complex, and after 60 minutes of cleavage reaction, you can see that the uncut double-strand DNA become 2 different fragments, fragment 1 and fragment 2. So based on this figure, you can conclude that our complex is specific and functional in target cleavage. You can also see the evolution of the DNA concentration over time. The uncut double-strand DNA start at 5 nanogram per microliter and finish to around 0.5 nanogram per microliter. Something that is also interesting is like we can evaluate the impact of some degraded Cas9 proteins. So we do different complex, this time with a non-stress Cas9, stress Cas9 2 weeks 30 degree, stress Cas9 with 4 weeks at 30 degree. And you can see that degraded Cas9 has a negative impact on the cleavage activity of the Cas9 RNP complexes. For example, for the non-stress Cas9, we have like 75% of double-stranded DNA substrate versus only 30% with the stress Cas9 for 30 degree. So basically with this assay, with the in vitro DNA cleavage, you can determine the functional activity of Cas9, and you can also assess the cleavage rate of RNP complexes. So then something, a parameter that is really important, it's the isoelectric print. And basically it's the case for all the proteins that we need to characterize. So this technique is a high resolution technique that separates species based on their pI, taking into account solvent exposure residues and internal amino acids. So here you can see we generate some profile for the 3 different Cas9 variants using the Maurice-C instrument. And you can see here we have on the corner, you can see the theoretical pI value. And you can see that we have a very good correlation with the experimental values. And so we have some key benefits for using this method, Maurice instrument. So basically we have a very short analysis time, only 25 minutes. This method requires a very small amount on sample, only 0.02 milligram per ml. And with this method, you can discriminate differences in the pI of different Cas9 variants. Now, I want to talk a little bit more about how we can use mass spectrometry to have some information on these Cas9 variants. So first of all, we can do something that we can intact protein mass measurement. So this method can reveal the heterogeneity of proteins and allow comparison of molecular mass. So here, again, we injected the 3 different Cas9 that we have, and you can see that we have one main peaks, and then we have a very small peaks before the main peak. So this is some impurities of the Cas9 proteins. You can see also that for Spike-Cas9 and SP-Cas9, we have the same molecular weight, around 162 kilodalton, and for AC-Cas9, it's a little bit smaller, around 127 kilodalton. But something that is also interesting is like when we do the deconvoluted spectra, so meaning we just do the deconvoluted spectra of the main peak, you can see that we don't have only one mass, but we have multiple masses due to different combination of PTMs. Something that it was interesting is like we observed that the difference between the peaks was plus 78, 178 Dalton, corresponding to some gluconolylated forms of Cas9. And this post-translational modification, it's commonly observed in the E. coli cell line, for example. Something that is also interesting is like we can do the distinction between Spike-5 and SP-Cas9, something that is really interesting because as I mentioned earlier, Spike-5 allowed to have a very low level of off-target. And you can see that we can exhibit a mass of difference of 85 Dalton. If you see the red dot, you can see that we have a shift of 85 Dalton between this peak and this peak. And it will be the same for all the different peaks. So basically with this method, it's kind of very interesting because it's suitable for batch-to-batch comparability study, but also as an identity method for Spike-Cas9. Now, if we want to go further, we can develop a peptide mapping for Cas9. So peptide mapping is conventionally used and recommended by the regulatory agency. With this method, it's used for proof of identity, primary structure characterization, and quality assurance. So in this method, we develop a specific method for Cas9 because Cas9 is not very stable. So we decide to remove all the unnecessary steps. So, for example, reduction and alkylation, since Cas9 don't have any disulfide bridge. We also prefer use lysine over trypsin in order to generate less fragments and to avoid to limit the risk to have some coelution of the peak on the chromatogram, especially if you want to use a UV detection. And with our method, we obtain a sequence coverage of 98%, and most importantly, we can capture 2 unique peptides that correspond to the mutation to distinguish the spike Cas9 from wild type. You can see on the chromatogram on the bottom that for spike in purple, we have only the peptide corresponding to the spike specific peptide, while for the Cas9, we have only the peptide corresponding to the wild type. Now, it's also important to show that this method is specific. For example, in the case of the manufacturer produce the 2 different variants at the same manufacturing floor, we need to be sure that if we have any cross-contamination issue, we should detect that with our method. And as example, you can see with the orange trace that if we do the mix PI5 and Cas9, we're still able to determine this cross-contamination issue since we observe wild-type peptide and spike Cas9 is for this sample. So, here I gave you a summary of all the different methods that we can use for Cas9. So we can use CSDS as a purity test. We can do in vitro DNA cleavage to have more information about functional activity of RNP complex. We can determine the PI value by ICIF. And of course, we can use a mass spec to have some information about Cas9 identity at the intact and peptide level. So in conclusion of this part, all these analytical methods are applicable to different Cas nucleotides, for example, Cas12, for example, and all these methods can help to establish specification that will meet the requirement of regulatory agencies. So for the second part of this talk, I want to discuss about RNP complexes and how we can have more information and evaluate the impact of guanine RNA or Cas9 on the RNP complex formation. So when now the current specification of the gene editing reagent, meaning Cas9 and guanine RNA, are set separately. For example, for Cas9, we know that we need to determine PTMs, aggregate and truncate, and for the single guanine RNA, we need to evaluate the impact of aggregate and secondary structure. But we don't have any strategy to evaluate the impact of the 2 different species in the formation of Cas9 RNP complex, and basically on the Cas9 RNP formation and stability. So it's for this reason that we try to develop some HPLC method in order to have more information on the Cas9 RNP complex formation. So first of all, we decided to develop a SEC method. So SEC is a reference method for the quantitative measurement of size variance. And here it was interesting. So basically we inject 3 different samples. One is only Cas9, the other one single guanine RNA, and the last one was a complex between Cas9 RNP complex, so meaning single guanine RNA plus Cas9. So if we observe the blue trace, you can see that we observe the Cas9 monomer with a molecular weight of 160 kilodalton, and you can also see the minor peak corresponding to the Cas9 dimer. Now, if we observe the pink trace, you can see that we observe the single guanine RNA monomer with a molecular weight of 33 kilodalton, so it's something that we expect. But we did not expect the other peaks at 8 minutes and at 7 minutes corresponding to single guanine RNA aggregate. And so we have a very high amount of aggregate. And this has a negative impact on the Cas9 RNP complex, because if we look at the orange trace, you can see that we still have the single RNA monomer, but also the huge amount of aggregate and probably also the formation of Cas9 single guanine RNA aggregates. So And something also that we can notice is like the Cas9 is completely converted into Cas9 RNP complex because you cannot see any Cas9 monomer peak on the orange trace of the Cas9 complex. So then we try to have more information on this presence of high amount of aggregate in the single guide RNA material. So in order to do that, we selected 4 different single guide RNA. So A1, B1, A2, B2. And for A and B, so it's referred to RNA coding different gene, while 1 and 2 indicate that there are some differences in the cRNA sequence. And so you can see that we have different profile, but at the end, we have the same species. So basically, we have our monomer between 40% and between 53% and 90%. And then we have a different aggregates. We identify dimer, but also trimer and tetramer. So for the tetramer, for example, using moles detection, we observe that this tetramer has a molecular weight of 145 kilodalton. And so we can conclude from this side that we have varying amount of single guide RNA aggregate, and also that the different aggregate formation due to the sequence specific of each single guide RNA. So then something that we also observe is that when we use a high temperature, basically if we do the denaturation step of single guide RNA at 70 degrees for 10 minutes, we can remove all the different aggregates and we can only have the monomer. And if you see the scale, you can see that the monomer, it's very we have a higher amount of monomer versus when we don't eat the guide RNA. And so basically we can conclude that we have a better purity when we eat the guide RNA. Something that is also interesting is like if we took the same sample and we inject the same sample after 6 hours, 12 hours, and 24 hours, we can observe that the single guide RNA monomer are very stable and they do not go back into aggregate form. So then the thing that we did is like we eat without eating the guide RNA, we add the Cas9, and you can see that we still have all the different aggregate corresponding to the single guide RNA, but we have also additional peak 1, 2, 3. And so then using moles detection, we can have a better idea about what is this different species. And so here, I'm sorry, but you cannot see very clearly, but basically the difference between 1 to 3, it's only the single guide RNA, because for the protein, we have the same molecular weight, meaning that we have only one monomer of Cas9, but between 1 to 3 is only the single guide RNA that change. So for the peak 1, we have the monomer, peak 2 dimer, and the peak trimer. So basically for the peak 3, we have a combination between the trimer guide RNA plus one monomer of Cas9. And so Cas9 RNP complex formation between Cas9 and single guide RNA aggregates. But now I want to show you one example when we hit the guide RNA. What happens? If we hit the guide RNA, we remove the peak 3 and the peak 2, and we have only the peak 1. And you can see that in this case, in this orange rectangle, you can see that all the spaces composed of monomer form of Cas9 and single guide RNA only. And why was it different peaks? It could be some different conformation between the different Cas9 RNP, or sometimes it could be also one PTM from Cas9. For example, deamidation can impact on the retention time with these HPLC methods. So something that I want to show you now is like what will be the difference in terms of cleavage activity if we if we use this complex without eating the guide RNA and when we eat the guide RNA. And you can see here that we did that for two different RNP, for RNP A1 and RNP B1. And you can see that when we don't eat the single guide RNA, after 5 minutes, we have 75% of double-strand DNA substrate. But if we eat the guide RNA, it seems that the cleavage rate is higher because after 5 minutes, we delete like 40% of double-strand DNA. And we observe the same thing with the RNP B1 with the same percentage, around 50%. So basically from that, we can first conclude that the Cas9 RNP complexes derived from eating single guide RNA are functional and cleavage the double-strand target DNA. And when we eat the single guide RNA prior to the formation of Cas9 RNP, leads to more efficient cleavage. And so why? So the explanation is coming from here. So basically, in order to prove that, we analyze the single guide RNA and then we do the offline fractionation of the monomer and the dimer, monomer and dimer of the single guide RNA. And we do the complex for each of them. And you can see that when we do the complex with the dimer, we don't have biological function. The biological function is only from the monomer, meaning that when we eat the guide RNA and we increase the percentage of monomer, it makes sense that we have a better on-target activity of our complex. So it's time for the conclusion. So in this part, I show that the formation of different single guide RNA contribute to the different Cas9 RNP complex formation. That eating the guide RNA can improve the single guide RNA homogeneity. That can also retain the biological function, and more important, can cleave the double-strand DNA at higher rate, and also that the Cas9 RNP complex could be impacted by the physical properties of the gene editing reagent. And through this presentation, the key message was that the understanding the factor that impact the Cas9 RNP complex formation could provide valuable clue to better control gene editing for different clinical applications. And so for my last slide, I want to thank all the people involved in this project, especially Mariam Moshref, Oyong Lee, Dayu Shen, and thanks to GenScript for inviting me for this talk. Thank you.